न्यूरोटॉक्सिन प्रोड्यूसिंग क्लॉस्ट्रीडिया। यहाँ पे हम क्लॉस्ट्रीडिया की सिर्फ उन स्पीशीज की बात करेंगे the toxin of which affect the nervous system of animals as well as of human beings. Now, our main concern will be on Clostridia botulinum. It is actually a gram-positive bacteria that appears purple in color and is in rod shape. They grow in the absence of oxygen, that's why they are anaerobic. They have the ability to produce spores, thus they can survive under harsh conditions, and they are motile. It means that they possess flagella. Now, Clostridium botulinum has the ability to produce neurotoxin botulinum. This toxin affects the nervous system and cause paralysis in human beings as well as in animals. If you talk about the lethal dose of this toxin, it is about 1.3 to 2.1 nanograms per kilogram in humans. So from here you can judge the toxicity of this neurotoxin. Clostridium botulinum is divided into 4 groups. These are the Roman groups 1, 2, 3 and 4. Now some strain of Clostridium butyricum and Clostridium barrettae, they also produce a neurotoxin botulinum. Now let's talk about Clostridium botulinum only. It causes foodborne botulism, infant botulism as well as wound botulism, as the symptoms of which may vary or they may share with each other. Now Clostridium botulinum produce heat-resistant endospores. They are usually found in the soil, and in the soil they are in the dormant stage. These spores allow them to survive under adverse conditions. इसके अलावा ये ऑक्सीजन के ट्रेसेस को भी टॉलरेट कर सकता है. It means that it can survive under microaerophilic conditions. This is because of the enzyme known as oxygen superoxide dismutase. This superoxide dismutase allow them to survive in the presence of oxygen traces as well. Now, if we look at the serology of this bacteria, then keep in mind that it has 8 serotypes from A to H, and these serotypes produce multiple toxins. Now, usually toxins are destroyed at 100 degrees centigrade, but these neurotoxins, they are not even destroyed at 100 degrees centigrade. They are also resistant to the degradation by enzymes which are present in the gastrointestinal tract of humans as well as of animals. Now, if we talk about the botulinum neurotoxins A, B, E, F and H, it causes disease in humans only. Now, A, B, E are associated with foodborne illnesses, and if we talk about only the serotype E, it is associated with fish products. Type C produces limber neck in birds. Type D causes botulinum in other mammals. These are the mammals other than the human beings. While no disease is found to be associated with serotype G of Clostridium botulinum. Now, if we want to identify these types of toxins, we have to use mouse bioassay. इसके अलावा अगर हम genetic level पे A, B और E, F को differentiate करना चाहते हैं, we can use a molecular technique known as polymerase chain reaction. Clostridium botulinum can be isolated on a selective media, that is tryptose sulfide cycloserine growth medium. यहाँ पे हम anaerobic conditions provide करेंगे for incubation. इसके अलावा Clostridium botulinum microaerophilic condition में भी culture पे grow कर सकती है if the oxygen is present up to 2%. Now, if we talk about the foodborne botulism, the patient may suffer from double vision, blurred vision, drooping eyelids, muscle weakness, and even dry mouth. If we talk about the wound botulism, again it affects the vision, drooping eyelids, but there is difficulty in speaking as well as in swelling, and this can lead to fatal paralysis. Infant botulinum. In infant, the symptoms will appear in the form of weak cry, difficulty in sucking during feeding, irritability, dryness, and even constipation. Now let's talk about the neurotoxin. If we talk about the purified neurotoxin protein, it is 150 kilodaltons in weight, and how it affects the nervous system. It affects the peripheral cholinergic nerve endings. The purpose is to block the release of neurotransmitters. So these toxins can increase in size by combining with other molecules up to the size of 900 kilodaltons even. So for dealing with Clostridium botulinum, we have to handle this pathogen under biosafety level 2 practices, because toxins A, B, C, D, E, they are pentavalent, and we have used them in vaccines known as botulinum toxide vaccines. इसके अलावा अगर पेशेंट को बोचुलिनम हो गई है, इन सच केसेस वी कांट गो टूवर्ड्स वैक्सीन. इसके लिए हम एंटी टॉक्सिन प्रोडक्ट्स पेशेंट में प्रोवाइड करेंगे. Keep in mind that toxins are actually antigens. They are the foreign agents. They are the immunogens. So our immune system will produce antibodies in the form of antitoxins to fight against those toxins.